Good morning, everyone. Am I audible? So my voice is reaching to you? If yes, just give me thumbs up. Namaste. Okay, good. Good morning, good morning. Sindhu, Varsika, Maheshwari, good morning. Dragon, good morning. Good morning. Kartika, Satya, Multi Thunder. So name is like good morning, good morning. So before going to good morning, so everyone is having good morning. You had your breakfast and all. So I'm good. How about you? So you people are doing great. Okay. But here I think a lot more students are interested in cricket, the IPL one, because most of the chat I'm reading, it's like who won the match, who defeated. Okay, good. So in the last class, what are the things we have discussed? Like we have discussed in a brief, like what is prokaryotic cells, what is eukaryotic cells, and a difference between the prokaryotic cells and eukaryotic cells. And the main difference, I think we can say, where are you from? I'm from India itself. Yeah, it has stopped. Now it's clear. Video is not coming. No, I think it's okay now, right? Just give me thumbs up if it is okay. Okay. Fine. Thank you. So in the last class we were discussing about what? So we were discussing about the prokaryotic cells and eukaryotic cells. So the major difference between the prokaryotic cells and eukaryotic cells is the boundary, or we can say the boundary line. So if there is a compartmentalization, if there is a boundary, then we can say that it's a eukaryotic. If there is no boundary, then we can say it's a prokaryotic. So today we will discuss something about what? Something about the plant cells and animal cells, a slight difference. Thereafter we'll directly jump to what? It's a prokaryotic cells and its a structure. So prokaryotic cells and its a structure. Okay, so let's talk something about the prokaryotic cells. Prokaryotic cells and its a structure. But before that, we need to somehow a brief differences between what? A brief differences between the plant cells and plant cell and animal cell. So a slight differences. So what are the first difference? In plant cell and animal cell, which one is normally bigger in size? So obviously the point goes to the plant cell. So plant cell is little bit bigger in size. So here we can say plant cell is somehow larger in size, as compared to animal cells. So animal cells is having usually smaller in size. Okay. Next one is we can say if you are coming from the outside to inside. Let's say I'm having the plant cell. I'm having the plant cell. I'm just starting staring at. So what are the things that is outer side? Or we can say what are the things that I can directly see this, or we can say visualize this. So the first thing that I am visualizing here is the cell wall. So this is the first thing which I am seeing here. It's a cell wall. But in this case, there is no cell wall, so we can say it's an absence of cell wall. Okay. So next one is we'll go to some other parts. If you are talking about the cell membrane, so who is having the membrane? So can we say that membrane is present in both, like plant cells as well as animal cell? Because membrane is the only part, or we can say cell membrane is the only part that is present in all living cells. You are having the membrane, cow is having membrane, bacteria is having membrane, fungus is having membrane. So like membrane is present everywhere. Membrane is present everywhere. After that, we'll move to the next one. That is, if we are talking about the cell organelles. Cell organelles means what is there in the plant cell that is not there in the animal cells. So if you are talking about the plant cells, the most important structure that we can call it as a plasmodesmata. So I think there might be a possibility that you are familiar with the word, that is a plasmodesmata. So plasmodesmata is like its main function, main contribution is to connect two plant cells with the help of their cytoplasm. Like this is the one plant cell and this one is the other plant cells, and these two plant cells are connected by what? So connected by this plasmodesmata. So can we say that plasmodesmata is a living connection? Or we can say it's nothing but a cytoplasmic connection. In other words, I am just writing it's nothing but a what type of connection? Living connection, or in other words, we can say it's a cytoplasmic connection. What is that? Cytoplasmic connection. While in this case, cytoplasmic connections or plasmodesmata is present or absent. So we can say it's absent. Okay. Next one is, which cells can photosynthesize, or which cells can provide you the food? So obviously my first answer is plant cells. So plant cell is everything. I am getting oxygen, I am getting the food, or we can say most of the things or most of the required things, it is coming from the plant cells. That's good. A lot more responses I am getting at the same time. It would be great to interact with the students. So directly we'll go to the chloroplast. So obviously chloroplast is present in the plant cell, or this chloroplast comes under the plastid, or we can call it as a plastids. So in this case that is a chloroplast is absent. Is absent. Okay. Next one is, what else we can add? So here one more thing we can add is if the plant is huge, like let's say it's a mango tree. So obviously it's a mango tree after 10 years. It becomes taller than you, or we can say if the size of the plant, it's bigger than you. So bigger than you means we can say that more than one cells are present, and these more than one cells are present, or we can say attached rigidly, or we can say compactly it is present. So how it is compactly present? So we can say with the help of one structure, that structure is known as middle lamella. Hi. So here we can say next one is middle lamella is present, or middle lamella is also known as plant cement. Middle lamella is also known as plant cement, which holds two plant cells together, or we can say it's nothing but a cement, like it's a bricks. So two bricks are joined together with the help of what? That is a cement. Like, have you heard about the Ambuja cement, Ultratech cement, ACC cement? So this is nothing but the middle lamella. So you can say that this is nothing but the middle lamella which holds two plant cells together. Okay. So in this case, the animal cell don't need such kind of rigidity. Don't read, yeah, Nagarjuna cement. That's good. You are having a too much knowledge about the cement. So here we can say that is a middle lamella. So middle lamella is absent in what? That is the animal cell. Why? Because animal cell doesn't require that much of flexibility. Sorry, that much of rigidity. So rigidity is not here. Rigidity is not here. That's why there is no need of middle lamella. Means obviously, can we say that our skin is flexible? Our skin is flexible. But here, if there is a middle lamella, we can't do like this. Or we can say we can't move like this. That's why the need of. So here we can say again the middle lamella is absent. Okay. Next one is which one is having the nucleus, plant cells or animal cells? So my answer is both is having the nucleus, but their presence, their locations, or we can say their size is totally different. If you are talking about the nucleus, so nucleus is present here, or we can say nucleus is present in the plant cell, but somehow it is towards the corner, or we can say it is present in the corner. Why? Because most of the space is occupied by the vacuole. So nucleus is getting very less space. So that's why we can say that in this case nucleus is present in the periphery, or we can say here the nucleus is elliptical type of nucleus is present. Okay, so nucleus is present, but here we can say it's not central. So the nucleus is what? So nucleus is peripheral. Peripheral. In this case we can say the nucleus is what? So here we can say central nucleus will be present. Here the nucleus at the center, or we can say central nucleus is present. So am I clear to this? Like till this point, the difference between the plant cells and animal cells. Okay, now we'll move to the next. So what's there? Directly we can jump to other differences. So other differences we can say that, have you heard about the vacuoles? So here we can say the vacuoles are present, and what type of vacuoles? So we can say it's a large but present in the center. So here it's a large central vacuoles are present, and obviously the occupancy of this vacuole is like too much. We can say approx 90 to 95% of space is occupied by the vacuole, or we can say most of the space is occupied by the vacuoles. Okay, but in the case of animals, vacuoles are present or absent. So it is there, but its size is less. It is there, but its size is less. So here we can say small, and fewer vacuoles are there. Okay. Next one is, which one is having more lysosomes, plants or animals? Plants or animals. So obviously, animals is having more lysosomes as compared to plants, because most of the lysosomal activity, most of the lysosomal activity is done by what? That is the vacuoles. Most of the lysosomal activity is done by the vacuole, so very less requirement of lysosome is there. So lysosome is there, no doubt. So lysosomes are fewer, but in this case, lysosomes are most. So more number of lysosomes, more number of lysosomes. Acha. One more thing, like if I'm asking which one is having the we can say rigid shape, or we can say it's a definite shape, plants or animals? So which one is having definite shape, plants or animals? So I think plant is having the definite shape, or we can say regular one, while the animal is having some of flexible, or we can say it's irregular shapes can be there. That's good, that's good. I'm getting a lot more answers, and that is quick answer. So this shows that you are studying seriously, or we can say you are focusing yourself towards the study, and that is good. Because when you are studying, just do study only. When you are playing, just play only. Okay. So anyway, like we'll move to the next part. Lysosomes is done. So after the lysosome, which one is having the rigid, or we can say regular shape? So here we can say it is having rigid, or in other word, it's nothing but the, while in this case we can say it's a irregular one. Here we can say it's a irregular one. Okay. Now next one is, which one requires more energy, or we can say more powerhouse, plants or animals? So I think I'm just teaching. So whenever I'm teaching, I need ATP. And from where this energy is coming? So obviously this energy is coming from the mitochondria. Have you seen any plant teaching you botany? Have you seen any plant teaching you botany? So I think the answer is no, right? So that's why the plant don't need much of mitochondria, because plant remain at one place. So the plant is not moving from here and there. So the consumption of energy is very less. So here we can say it's a mitochondria is there, but its number is less. So less number of mitochondria, while in this case more number of mitochondria is there. Okay. Next one is which cell is responding fast, or we can say whose response is fast, plant cells or animal cells? So I think it's if you are asking some question to the animals and plants, so which will give you the fast answer? So did you ask any question to the plants? And have you ever had the answer from the plants? So I think obviously not. So animal will give you the fast answer. So if it is a human, the language is the answer. If it is a cattle, or we can say cow, and so it's a response will be, it's a we can say activity. It's a activity. So here we can say fast response to stimulus, and here it's a slow response to stimulus, and here it's a faster response to stimulus. So fast response to what? Fast response to stimulus. Okay, so these are the differences. Next one is plant cells and animal cell. Which one is dividing, or we can say which one is increasing its number by division? So I think in both the cases In both the cases, it will divide. It will divide. So animal cells as well as plant cells, both will divide. Or we can say both will increase its number. Both will increase its number. So here we can say cell division is there in both the cases, but the cell division in this case, or we can say in the case of plants, cell division, or we can say whenever the cytoplasm is dividing, this process is known as cytokinesis. And what are the methods we are using? So we are using the cell plate formation. That we will discuss later, how the cell plate is formed, because this is the plant cell. And plant cell is having what? That is a rigid cell wall. So this rigid cell wall cannot be changed. This rigid cell wall cannot be changed easily. So instead of changing this cell wall, we are just making one more wall in between them, so that it will divide into the two compartments. So here it's a cell division is there, and the cytoplasm division is known as cytokinesis. And this cytokinesis, one process it is following. The name of the process is what's that? It's a cell plate. It's a cell plate formation. Next one is in this case, if you are talking about what? That is the animal cells. Animal cell doesn't have any cell wall. So if it is not having any cell wall, what will happen? Can we squeeze it? Can we press it? So that the two compartments will form. And this process is known as what? It's nothing but the furrow formation, or we can say one groove is forming. Okay. So here we can say cell division is there in both the cases. Cell is dividing in both the cases, and that too during what? During the two cell, or we can say the cytoplasm division. So the division of cytoplasm is known as what? Cytokinesis. And this cytokinesis is done by what? So this cytokinesis is done by what? That is a furrow formation. Furrow means what? It's a groove. So next one is, what's the doubt? Veera Siva Reddy. So what's the doubt? Yeah, obviously the exam will be conducted. So whatever the syllabus we have discussed. Yeah, from the last week's syllabus. What does your doubt is there, but it's not related to this part. So we will discuss later. Like gibberellin, or we can say PGR, whatever that parts is there. So it's not related to this. So slowly we'll move, stepwise we'll move, so that it becomes easy for students to grab the concept. So now next one is what else we can add. So in this case, we can say that one is anestral and one is amphiestral. I am just briefing you what is anestral and what is amphiestral. So astrals are nothing but the what? It's like a spindle fiber. Or we can say, did you watch Spider-Man movie? Did you watch Spider-Man movie? Okay. So Spider-Man, whenever a Spider-Man is jumping from one building to other building, what will happen? Just it will do like this. and a lot more strings, or we can say that is a web-like structure is coming out. So with the help of this web-like structure, it will grab the surface, or we can say it will hold the building. It will hold the building. In this case also, the animal cell. The animal cell is having what? Centrioles. The animal cells is having the centrioles, and centriole is like what? Centriole is like a Spider-Man. So whenever the centriole will put their hand like this, so a lot more strings will come out. And that strings is nothing but the spindle fiber. And that is known as asters, or we can say astral layers are formed. Okay. So here we can say usually centrioles are absent. Next one is here, it's a centrioles are present. Okay. So next one is what else we can add. That is, usually centrioles are absent. Here it's a centrioles are present. So what type of mitosis? So here we can say the type of mitosis is anastral mitotic division, or we can say without string, without this wave, it is dividing into cells. While in this case, it is dividing with what? It is dividing with the help of asters, or we can say it's a help of webs. That is a Spider-Man. Okay, if the question is coming, like who is having a Spider-Man? The plant cells or animal cells? During mitosis. During mitosis, which one is having the Spider-Man? Plant cells or animal cells? So animal is having the Spider-Man, or we can say animal cell is having what? That is a centriole, and centriole is forming the web, and this web will help in what? Will help in pulling the chromosome, or we can say dividing the chromosome, or we can say dividing the cytoplasm. So many other things are there. Okay. So here we can say it's anastral, or we can say absence of asters, and here we can say it's nothing but the asters are formed, and that too is nothing but the amphiastral type of mitotic division. So these are the few differences we can say between the plant cells and animal cells. So just will recap what are the differences we have seen till this part. So I think little bit differences are there. So we have started with what? So we have started with the larger in size, or we can say size wise. Larger in size, one is small in size. Cell wall present, absent. Plasmodesmata present, absent. Next one is chloroplast present, absent. Middle lamella present, absent. Nucleus is peripheral, while in this case nucleus is that is a central. Why? Because here it's a vacuoles are present, or we can say most of the spaces are occupied by the vacuoles. That's why here it's a peripheral and this one is central. So here it's a large central vacuoles are there, and here it's a small and very few. Next one is the lysosomes are fewer. Why? Because the function of lysosomes can be done, or we can say can be helped. Can be helped by what? Can be helped by the vacuoles also. In this case, vacuoles is very less. One question I'm Meghna ready, that is, what is amphiastral? See, amphi means what? It's a two. Amphi means it's a two, and astral means what? It's like the web-like a structure. So in the animal cell, two Spider-Mans are there. So from both the pole, they are just producing, or we can say emitting the asters. So that's why we can say what? That's why we can say it's amphi astral. And in this case, there is no astrals, so we can say it's nothing but the N astrals. So did you get my point, Meghna? Okay. So now we'll move to the next one. So what will that web-like structure do? So web-like structure is, it's like a string. With the help of a string, I am just what? Holding something. Same as it is with the help of web, it is just pulling the chromosome. Like let's say these are the chromosome. One chromosome is pulled by this web and other is pulled by this. Other is pulled by this. And next one is a plasmodesmata. So plasmodesmata, main function of okay, yeah, obviously Spider-Man don't know. So here we can say the plasmodesmata. This is the one cell, this is the other cell, but these two cells don't have any connections. But I made some connections. So this is nothing but the what? This is nothing but the plasmodesmata. And with the help of plasmodesmata, what? That is the things are moving from this place to this place. So this is the main cytoplasmic connections. Okay. So did you get my point? What is the usage of plasmodesmata? Okay. So now we'll move to the next. Is let's talk something about the prokaryotic cells, or we can say that is a structure of prokaryotic cells. So till now we have discussed a little bit about what? Little bit about the prokaryotic cells, eukaryotic cells, plant cells, animal cells, animal cells. Now we'll start with the prokaryotes. So prokaryotes means what? What are the first thing that comes in your mind whenever you are hearing prokaryotes? So whenever you are hearing this prokaryotes, so yes, membrane is absent. Good. Primitive. Next one is, okay, well defined. And what are the organisms that firstly visualize? Or we can say, what are the organisms that comes before your eyes first? Whenever you are hearing this, that is prokaryotic cells. So unicellular, that's good. Unicellular, can we say it's a bacteria? Yeah. I'm just getting one answer, Shravani. That is bacteria. Russell, bacteria. Yeah, good. Bacteria. No, virus is not prokaryotes. Virus is not prokaryotes. Or we can say virus is acellular, means the virus is not having anything in its own. So it is totally depends on you. Like you are providing the energy. So you are providing good food, like whenever you are having the cashew nut or many other things, or we can say idli, dosa, sambar. So to whom you are, for what you are eating. So you are eating for the virus. If virus has entered in your body, virus is entered in your body. So that's why virus is not considered to be as a cellular. Okay. Euglena is not prokaryotic, Varsika. So euglena is eukaryotes. Euglena is eukaryotes. One is Risvita, that is good. Monera kingdom, like histone protein is absent. Monera, kingdom Monera. Whatever the organisms placed in the kingdom Monera is considered to be as a prokaryotes. In animal cells, gamete division, yes. In animal cells, or we can say in animal cells or plant cells, that is for the gamete formation, meiosis is required. So meiosis is required for what? So here we can say The blue-green algae, a lot more answers are there. So directly we'll start with what? That is a prokaryotic cells. So prokaryotic cells in this case, so firstly we'll write what? Prokaryotic cells. And you already have told a lot more answers like how prokaryotes looks like. Prokaryotic. Protozoan is not prokaryotic cells. Protozoan is not prokaryotic cells. Protozoans come under, yes, protozoan come under protista, and that protista is nothing but the eukaryotic. Okay. So next one is in this case prokaryotic cells. What are the things that is involved in the prokaryotic cells? So in the prokaryotic cells, few examples I am writing. So prokaryotic cells includes what? So includes, first is bacteria. First one is, or we can say it's a common, is bacteria. Is all unicellular organisms are prokaryotic? No. Eukaryotic cells can also be the unicellular. So unicellular and prokaryotes and eukaryotes are different thing. So unicellular and multicellular, that is based on the number of cells. That is based on what? Based on the number of cells. While in the case of eukaryotes and prokaryotes, that depends on whether it is having the membrane bound organelles or not. Whether it is having membrane bound organelles or not. So here it's a includes, one is bacteria, or we can say in the eubacteria, one more is coming, eubacteria, that is we can say it's a blue, green algae. So, can anyone answer, without blue-green algae can you survive? So how many of you can survive without blue-green algae? How many of you can survive without blue-green algae? So obviously there is a big question mark in the absence of blue-green algae on the survival. So no one can survive. And why no one can survive? Because let's say this is the blue-green algae. And this blue-green algae is only have the capability, or we can say the potential to fix the nitrogen from where? To fix the nitrogen from the atmosphere and transfer into the soil. After that, this nitrogen is utilized by what? That is the plants. And this plant can utilize this nitrogen to form the amino acids, proteins, vitamins, and many other things. And these nitrogen, this nitrogen which is there in the atmosphere now becomes the part of plants. Cause of what? Cause of this cyanobacteria, or we can say it's a blue-green algae. After that, this fruits, or we can say the leaf, or whatever the parts we are having from the plants, this is the best source of nitrogen. So we are taking nitrogen from where? Plants. And plant is getting the nitrogen from where? The cyanobacteria. And cyanobacteria is getting the nitrogen from where? That is the atmosphere. but we can't directly catch the nitrogen from the atmosphere, or we can say fix the nitrogen from the atmosphere. Okay, so that's why we should be thankful to what? We should be thankful to the cyanobacteria. So every day whenever you wake up, just say thankful to blue green algae. I'm just gratitude, or we can say I'm highly obliged to you. Who? That is a blue green algae. After that, here we can say it's a archaebacteria. Obviously, this thanks blue green algae. That's good. Hasni, that's good. Well, like one should be gratitude for the things. So here it's archaebacteria. So who has to pay attention to this archaebacteria? Like archaebacteria is useful to directly to us or to someone else? So I think it's someone else, this archaebacteria. Like archaebacteria is doing something for the cattles. Like we can say it's a cow and buffalo. These two things I know in Telugu. Like cow is ow and buffalo is bare. Okay. So here it's a cow and buffalo. So cow and buffalo is eating what? Eating on the cellulosic material. Eating on cellulosic material. But this cellulose cannot be digested by cow and buffalo. This is not digested by the cow and buffalo. For that, which one comes? That is a archaebacteria will come. Okay. So what's the doubt? Rishrita? Okay. So here what I said, archaebacteria is doing help, or we can say doing favor for what? Doing favor for one question is, what is difference between the algae bloom and cyanobacteria? See, bloom means what? Bloom means excessive growth of something. Excessive growth of something is known as bloom, and it can cause due to the algae. So that's why we can call it as a algal bloom, and this cyanobacteria can cause the algal bloom. It can cause the algal bloom because whenever you are going to the lake, or we can say pond, and if you are not using the lake and pond frequently, then what will happen? The whole water turns into green. The whole water turns green, and that greenish part is due to what? It's a cyanobacteria, or we can say it's a algal bloom. So bloom can be of anything. Okay. So are you getting my point? What is bloom? So now we'll move to the next one. Is this includes also, that is nothing but the mycoplasma. And mycoplasma, or we can say it's nothing but a piplo, or we can say pleuro. Obviously water bloom is bad for us. Water bloom is bad for us because it will pollute the water. Because it will pollute the water, that's why it's a bad. So piplo is nothing but the pleuro, pneumonia like organism. So this also comes under this part. So mycoplasma, or we can say it's a pleuro, pneumonia like organism, or we can say it's a piplo. So the last two one, mycoplasma is not having the cell wall. So mycoplasma is not having the cell wall. This is the difference, while all other is having what? The cell wall. So here we can say the mycoplasma and whatever it's a piplo is there. This is not having what? This is not having the cell wall. So here we can say it's a lack of Lack of cell wall. Okay, next one is that complex in structure or simple in structure? So I think it's behaviorally, it's a complex. Behaviorally, it's a complex, but whenever you are looking, it looks simple. So it looks simple, but behaviorally, it's a complex. So here we can add few points like how it looks like. So the first point we can say is structurally simple, but behaviorally it's a complex. But behavior wise complex. But behavior wise it's a complex. Okay. So after that, what else we can add? So here we can say the structure, or we can say the shape of this bacteria is similar or variable. Yes, can survive without oxygen. That is a mycoplasma. Mycoplasma can survive without oxygen. But it's not like that. Whenever oxygen is present, the mycoplasma will not survive. So generally, normally, in the presence of oxygen, mycoplasma is surviving. But if there is no oxygen, or we can say lack of oxygen, then also it can survive. So in other words, we can say even in the absence of oxygen, mycoplasma can also survive. Even in the absence of oxygen, mycoplasma can survive. So directly we'll move to the next part. So next part is the shape of bacteria. Can you tell me, like, the shape of bacteria can be of mainly 4 types. Mainly of how many types? That is 4 types. And these 4 types are, so the first type is, we can say it's a coccus. First type is coccus. Next one is bacillus. So one is coccus and one is bacillus. And next one is, sometime it is nothing but a vibrio. Other time we can say it's nothing but a spiral shaped. Other time it's nothing but a spiral one. Or we can say it's nothing but a spiralum. Okay. So here, if you are properly seeing the shape of this, so the coccus is normally what? It's a spherical. It's a spherical, while in this case, these are the bacillus, while in this case, it's nothing but a comma shaped. Next one is, it's nothing but a spiral. Okay, so obviously each one is looking different from others. So based on the shape of the bacteria, we can divide. So it is nothing but a spherical. Next one is, it's nothing but a rod shaped. Here it's nothing but a. Next one is a spiral, or we can say it's nothing but a helical in. So just we'll look into few examples. So the best example for the coccus is, can you name some coccus? Chalo, theek hai, I'm just giving you a hint. That is bacteria, or we can say a coccus which can cause pneumonia. That is a streptococcus. Good. Streptococcus, Staphylococcus. Okay. So we can say Diplococcus, Streptococcus, Staphylococcus, and at the same time, one is methanogens, or we can say in methane it's there. So Methanococcus. So a lot more examples can be there, Micrococcus, but I'm just writing few examples. Few examples is nothing but the, if it is two in number, we can say it's a Diplococcus. Next one is when it is present in a single chain, like more than one is there. So we can say it's a Streptococcus. Same as it is, we can say it's a Methanococcus. So a lot more example we can have. Okay. Now just tell me something about Bacillus, the best example which can convert Palu into Pergu. Which can convert Palu into Pergu. That's great. Lactobacillus. Lactobacillus that converts Palu into the Pergu, right? These are the name I know. Same as his anthrax, like Bacillus anthracis. So Bacillus anthracis. And next one is an organism which is commercial, but it can cause what? It can cause cholera. That is a Vibrio cholera. Good, Henry. So here it's a Vibrio. Next one is nothing but the, here it's a spiral. So we can say it's a Treponema palladium, or we can say spirillum. A lot more examples can be there. So here it's a, so these are nothing but the based on what? Based on the structure, or we can say based on the shape, this can be of different types. After that, we'll move to the next, like how it looks like. What is the structure of this? What is the structure of this? That we'll talk here in the class. So firstly, I will just draw the diagram. Here I will draw the diagram, and in the next class, we'll discuss the point by point. Okay. So firstly, if you are moving to the, so let me firstly see, is there any doubt? Any doubt from this part, so that I can deal? Okay, good. So, what's a doubt? Donapati. So, most of them is not having any doubt. But whoever is having doubt, just write there your doubt. Okay, no one is having. So directly I'm just I'm drawing the diagram. So firstly, how many of you have seen the bacteria by naked eyes? How many of you have seen the bacteria naked with the help of naked eyes? So exam time, or we can say whatever the syllabus, so everything will be uploaded. Don't worry about that. So be stick with the syllabus. That's all. So anyone who has seen the bacteria with the naked eyes? So why it is not possible to visualize the bacteria with the naked eyes? Can you tell me? Because it's a microscopic in nature. It's a microscopic in nature. But if you are having a microscope, obviously it's a visible. Obviously it's a visible. But to study the bacteria, and that too is nothing but a microscopic, means I am not able to visualize this with the help of naked eyes. So firstly, or we can say bring one microscope, and with the help of the microscope, we can visualize. But before visualizing this, we need to stain it first, make it colorful. Firstly, make it colorful. So let's say I found one bacteria. And firstly, you have to make it colorful. Then just put it under the microscope so that you can properly visualize this. Okay. So that's why it requires, or we can say study of bacteria requires what? Study of bacteria requires staining techniques. And one more is there, that is nothing but the gram stain. Who is gram? Can anyone tell me who is gram? Gram. So who is gram? Yes, Gram is the name of the scientist. And if you are talking about the Gram, so Gram was the name of scientist, and he started observing the bacteria. So obviously he has to put some dye, or we can say firstly they have to make it colorful. So that is known as staining. So Gram is staining. It can be positive, it can be negative. So if it is according to the Gram, then it's a positive. If it is against the Gram, this is negative. So the Gram positive bacteria and Gram negative bacteria. So according to Gram, how many type of bacteria is there? According to Gram, how many type of bacteria is there? So Okati or Rendu? So obviously it's a 2, right? So one is Gram positive bacteria and one is Gram negative bacteria. That's good. So in the next class, we'll talk something about what? We'll talk something about the prokaryotic cells, and that is the structure of prokaryotic cells and what are the functions of each and every part of the prokaryotic cells. Okay. So we'll meet you again. So till now, bye bye everyone.